So you should have already looked at both passive and active transports. And as we've seen with both passive and active transport, or diffusion, right? Diffusion in terms of when we're dealing with passive transport, and pretty much we're able to move large amounts of materials into and outside of the cell. The only thing is with these little materials, right, we're looking at pretty much dissolved particles, right? So we were looking at dissolved particles, right? We're looking at things like glucose molecules. We're looking at various ions like potassium and sodium ions. We're looking at molecules like water, carbon dioxide. And the thing is, what happens, right, when we're wanting to move a large amount, right? Or should we say a bulk amount of things into or outside of the cell? So we've got a process called bulk transport, right? Which pretty much very similar to active transport. Bulk transport also uses ATP as their source of energy. Now, there are two forms of bulk transport. Two forms, and let's use a color. So, two forms, and these are endocytosis and exocytosis. Let's look at endocytosis right now. So endocytosis, pretty much, there are really two forms of endocytosis, and those are phagocytosis right, and this is pretty much what we call cell eating, and pinocytosis, which is pretty much cell drinking. But both of them refer to endocytosis, things that are pretty much coming into the cell. So these are particles that are outside of the cell that are coming into the cell. Now, in terms of phagocytosis, right? Phagocytosis is like cell eating. So let's draw here. We're going to draw our cell membrane, and we're going to focus on phagocytosis. Phagocytosis. So we've got our cell membrane, all right, and I'm not going to. Not going to draw it as a. Actually, you know what? Let's draw it as a thin, just to show that it's a bilayer, a phospholipid bilayer. So here's the cell membrane. Now the thing is, with this cell membrane, we have some kind of a food particle, right? So here's a food particle, right, outside, right? So that's food. Let's say food for the cell. So what happens now with phagocytosis, the cell membrane, right? The cell membrane is going to pretty much extract, so to speak, parts of itself, right? The cell membrane forms what we call these false feet. They form pseudopods. You probably came across the term pseudopods when talking about single-celled organisms like the amoeba. So what they do is they form these pseudopods, which are like fake arms around, right, around the food particle that they're trying to bring in. So what happens is they engulf, right? They engulf and they engulf this food particle, right? They engulf this food particle and eventually bring it inside, right? They bring it inside the cell. So the pseudopods pretty much close up and form a vesicle, right? Which now this is, let's say, the part 2 to this diagram. And let me erase some of this, right? Because this is what's going to happen, right? It's going to engulf, right? And all of a sudden, yes, of course, the cell membrane will get a little bit smaller, right? Because now this is what we call a phagocytotic vesicle, right? So it kind of forms this membrane-type vesicle that eventually will make its way towards some kind of a lysosome, right? And it'll pretty much bind to a lysosome, because that is where digestion of this particle will actually take place, right? So the phagocytotic vesicle will bind to this lysosome and eventually digest this piece of food. Now, nutrients formed by this kind of digestion moves through the vesicle's membrane and into the cytoplasm, where they pretty much get released to the various parts of the cell that are needed. Now, white blood cells, right, also do something similar. They also engulf invading bacteria using phagocytosis, removing them from the bloodstream and other body tissue. Very similar to the way this phagocytosis is. But now, if we're going to look at now a different, right? We looked at phagocytosis, but what is pinocytosis? Pinocytosis, very similar to that of phagocytosis. The only difference is we have smaller particles, right? There are smaller particles that are going to make their way into the cell, right? So pretty much pinocytosis, right, again, will form these pseudopods, right? Will form pseudopods right around, engulfing these particles and then forming not a phagocytotic vesicle, but a pinocytotic vesicle, right? So pretty much cells will now bring like cholesterol molecules into their cytoplasm for where they're needed, right? And pretty much this is a way of bringing in smaller particles, this cell drinking, right? And now if we want to erase, let's say, this particle here to represent what's really in here, well, we have all these little tiny particles that, again, will make their way to wherever the various parts of the cell are. Now, we looked at endocytosis, and this is where we were bringing things into the cell. Let's look at something called exocytosis. So, with exocytosis, we are trying to remove things from the cell. Now, we're now using exocytosis. Cells can move large amounts of material outside into their cytoplasm and eventually make their way outside of the cell, right? So cells produce substances like enzymes and hormones that need to be exported out of the cytoplasm and out to outside the membrane. So what they form is they form these type of secretory vesicles. Right? So they form these secretory vesicles because they're trying to secrete something, right? They're trying to secrete something that they have produced. So what happens is these secretory vesicles, they will attach themselves to the cell membrane, right? So here's our cell membrane, and here is just like in phagocytosis, but now we are making our way outside. So we are trying to bring out some kind of a particle, right? Let's say it's something like this. And we're trying to exit this, right? This is trying to leave. So what happens is this is going to make its way towards the membrane, right? It's going to bind itself to the membrane. It's going to release, right? It's going to release this particle, right, that they're trying to get rid of. Let me get rid of this. Right? It's going to release the particle, and then all of a sudden, remember how using endocytosis, right? Using endocytosis, we said that the membrane got smaller because part of the membrane became this vesicle and went in. And this is a way in which now this vesicle, this secretory vesicle, will now fuse to the membrane, and pretty much kind of it goes back and forth to kind of keeping the membrane the same size. So all of a sudden this membrane is going to stretch out and pretty much just remain its actual size, and it's just kind of gotten rid of the particles that they need. Now, one example of what is this kind of particle? Well, this kind of particle could be some kind of a protein that has just been made, right? So now let's look at how this protein gets made using the structures that we know in a cell. So if we look at the cell, we have proteins that enter the endoplasmic reticulum. So we're going to have the endoplasmic reticulum. And we're going to have our ribosomes, right? Because this is the rough endoplasmic reticulum. This rough endoplasmic reticulum is where these proteins pretty much get moved. So they enter into the endoplasmic reticulum, and proteins are going to be made by these ribosomes. So these are the ribosomes. I shouldn't, right? These dots here, right? This here, that's a ribosome, that's a ribosome, and this here is the endoplasmic reticulum, but it's considered the rough endoplasmic reticulum because they have ribosomes. So they get created here, and what happens is this protein makes its way, right, because the rough endoplasmic reticulum is a passageway. It's kind of like a transport system. It's going to make its way to something called the Golgi apparatus, right? And these are the Golgi apparatus, or sometimes they're referred to as Golgi bodies, right? So they're as Golgi bodies. So all of a sudden now, this particle here is going to make its way into the Golgi bodies, right? Where from here it will form a vesicle right? Form a vesicle that is going to carry this, right? So now this here is what we refer to as our secretory vesicle. And what's going to happen is it's going to make its way towards the cell membrane. So it's going to make its way towards the cell membrane, where it's going to bind to the cell membrane. Right? It's going to bind to the cell membrane, allowing this particle to escape or to leave, right? So this protein is now secreted into the extracellular fluid, right? This is the outside of the cell, to go to wherever they are actually needed. And now this secretory vesicle is going to become a part of this membrane. Of course, it doesn't change color, but it's just to show you that part of the membrane from the Golgi bodies, or part of the membrane from the secretory vesicle, fuses itself with the cell membrane. And this is pretty much a typical exocytosis that actually occurs within our cells.